Hello class 10th. Hope you all are good. Now students, today in this video I am going to continue the same chapter, that is our chapter number 1 in biology, the cell cycle, cell division and structure of chromosomes. In this chapter we will today discuss about the significance of mitosis and the difference of mitosis in plant cell and animal cell, and what is meiosis and its significance, right, and structure of chromosomes. Let's start the chapter, the cell cycle, cell division and structure of chromosomes. Significance and importance of mitosis. Now, mitosis help in growth, repair and regeneration of body, cells, tissue and organs. Mitosis help in growth because all of you know life begins with one single cell, then divided into 2, then 4, then 8. In this way the body of an organism is formed. It also help in repairing the worn out tissues and or cells. Now third is your regeneration. All of you know what is regeneration. Sometimes you have seen just like if the tail broken tail of a lizard, it can be regenerated. To regenerate, actually the lost part maybe these are organ or tissue in if they are reformed, and these are reformed just because of mitosis. Now next is your second importance in mitosis also help in asexual reproduction in unicellular organism, right? Like amoeba, how the amoeba reproduce? Simply one amoeba divide into two parts, and both the parts they are called daughter amoeba, and then they grow into new individuals. This is called what? Mitosis. So mitosis also help in asexual reproduction in unicellular organism. Mitosis keeps or maintain the number of chromosome equal and constant in all the body cells of an organism. All of you know what is mitosis. Mitosis is a type of cell division in which a cell divided into two cells, two daughter cells, and each containing same number of chromosome as present in the parent cell. Means daughter cell having same number of chromosome, right? That's why in this way it maintain the number of chromosome equal and constant in all body cells of an organism. Next is your differences in the mitosis in plant cell and animal cell. In animal cell, during mitosis, what's happened? Two esters are formed. Why? Due to presence of centrosome. But in plant cell, no esters are formed as no centrosome, right? There is no centrosome. Now next is your cytokinesis. Takes place by the constriction or furrow in the cytoplasm. What is cytokinesis? Division of cytoplasm. But in plant cell, what's happened? It takes place by the formation of cell plate. Just look at here. After just telophase, just look at here. Okay, telophase, what's happened? This constriction of furrow takes place, means the cytoplasm is dividing from outside to inside just due to the formation of this furrow, right? But in plant cell, this is the last stage, telophase. Here what's happened? The cell plate is formed. It is a cell plate is formed, and which start from center to periphery. Periphery means from outside. Understood? Achha, how you will differentiate between a plant cell and animal cell? This is your animal cell, this one is your plant cell. Plant cell, if suppose in question plant cell is given, how you will identify? Plant cell is always having a two membrane. It has always a double membrane. Outer one is your cell wall, inner one is your cell membrane. But in animal cell, there is only one cell membrane. Understood? Okay, now next is your third point is your cytokinesis proceed from outside towards the center, that I just showed you. And here in plant cell, cytokinesis proceed from center towards the periphery. Periphery means to outside. Next is your spindle fibers are formed between two esters.
means what's happened, the spindle fibers are formed between two esters, because in plants animal cell esters are there, but in plant cell spindle fibers develop from the cytoplasmic strands which are called microtubules. And last point is animal cell spindle formed is amphiestral. What is the meaning of amphiestral? Actually the spindles has two esters, because all of an animal cell esters are formed and spindles are between two esters, so it is called amphiestral. While in plant there are no esters, so what is anestral? Anestral meaning the spindle has no ester. Understood? So in plant cell spindle formed is anestral, right? Now come to meiosis. Meiosis is a type of cell division in which cell undergoes two successive division producing 4 daughter cells, each containing half number of chromosome as compared to the parent cell. It is another type of cell division which generally takes place in reproductive cell. And in this type of cell division, it is followed by two successive division, in which how many daughter cells are formed? 4 daughter cells are formed, each containing half number of chromosomes, right, as compared to the parent cell. It is also called reductional cell division. Why? Because number of chromosomes become half in the daughter cell. Next is your meiosis takes place during gametogenesis. What is gametogenesis? Means during the formation of gametes in reproductive cell. This process meiosis takes place. It means this process takes place during the formation of sperms and ova in animals and human beings, while in plants during the formation of pollen grains and ovules. This meiosis cell division takes place. Now next is your significance of meiosis. Meiosis help in formation of gametes, right, and help in variation and evolution. What is variation? Variation is actually what there's always the differences among the between the offsprings of the same parent. That differences are called variations. All of you are a little bit different from your brother and sister, so these differences are called variations. It also help in variations and evolution. Meiosis also help in maintaining chromosome number constant, right? Now just remember the stages of meiosis is not in your syllabus, so you will only study about meiosis this much only, definition and its significances, right? Okay, now next is your difference between mitosis and meiosis, right? Mitosis occurs in somatic cells, right, and meiosis occurs always in reproductive cells. Mitosis produces 2 daughter cells while meiosis produces 4 daughter cells. Mitosis completed in 1 division, meiosis completed in 2 successive division, right? And next is your full set of chromosome is passed on to each daughter cell in mitosis, because all of you know that the daughter chromosome has same number of chromosome as present in the parent cell, while in meiosis only half the number of chromosome is passed on to each daughter cell. That means daughter cell having half number of chromosome present as present in the parent cell. Now the question is, what is chromosome? Chromosome is a thick rod-like structures bearing genes present in the dividing nucleus, and the number of chromosomes always remain constant in the cell of each species. All of you know in human beings, how many number of chromosomes are there? There are 23 pairs of chromosome, right? Depending upon the number of chromosomes, the cells are said to be haploid or diploid. What is haploid cell? A cell in the nucleus of which each type of chromosome is 1 in number, right? And diploid cell is a cell in the nucleus of which chromosomes are present in the form of pairs. Now next is here, what are homologous chromosome? 2 chromosomes of 1 pair which are exactly similar and bear same genes at same loci, means same position, then the chromosomes are called homologous chromosome. Okay, now next is your structure of a chromosome. Chromosomes are highly condensed chromatin fibers formed at the prophase stage of cell division. The structure of chromosome is best studied either during metaphase stage or anaphase stage of cell division. Actually, chromosomes, they are thin, long and thread-like structures consist of two identical strands called sister chromatids. And two chromatids, that is your sister chromatids, they are joined together at a point, and this point is called what? Centromere. So what is centromere? It is a point on chromosome where the two chromatids of each chromosome are attached. And parts of chromatids on the two sides of centromere, they are called arms. In each chromatid is present a highly coiled threads of DNA and proteins, and but they are called chromonema. Chromonema contains number of bead-like structures along its length, which are called chromomeres. The chromonema is surrounded by matrix, which is covered by a covering that is called pellicle. A chromosome consists of the following regions, that is your first is your primary construction, and primary construction is also called centromere. It is a meeting point of two arms of a chromosome, and this is the reason where spindle fibers attach to chromosomes during cell division. In some chromosomes, secondary construction or nuclear organizer is also present at any point of the chromosome, which are responsible for formation of nucleolus in the nucleus. Next is your telomere. Telomere is actually the end of the chromosome, or the terminal region on either side of the chromosomes are known as telomere. Actually, each extremity of chromosome has a polarity that prevents it from joining adjacent chromosome, and it also maintains the and provides stability to the chromosomes. Some chromosomes, they possess a small fragment or knob-like structures at one end of the chromosome, and they are called satellite. Such chromosomes are called as the SAT chromosomes, means SAT chromosomes. Chemical analysis of chromosomes shows two types of nucleic acids, that is your DNA and RNA. And actually each chromosome consists of about 40% DNA and about 60% histones, which is a special kind of protein, and non-histones and certain metallic ions. The histones and DNA form a kind of complex in which the DNA strands wind around a core of 8 histone molecules, and each such complex is called nucleosome. A single human chromosome may have about 1 million nucleosomes. Okay, thank you children, and rest of the part of this chapter, that is your structure of DNA and genes and different types of chromosomes, I'll explain in the next video.